Good morning, everyone. Good morning. Whether I'm audible? Yes. Good morning. Whether I'm audible? Yes. Whether I'm audible, kindly just reply whether I'm audible. Yes, yes. Yeah. Today we are going to just revise plant tissue culture, okay, then techniques in biochemistry and biotechnology. Apart from that, we are going to see about nitrogen metabolism, okay. These three are very important topics, okay. Very important topics, okay. Why means this techniques in biochemistry and biotechnology. From here, sure you will get minimum 4 to 5 questions, okay. This is the deciding factor for getting top 5 ranks, because some of the peoples will not read about the techniques, okay, because of the difficulty they will leave that one, okay. But every year they will ask 3 to 4 questions from these techniques in biochemistry and biotechnology. So this is the most important topic, okay. Next most important thing is nitrogen metabolism. Minimum 3 to 4 questions they will ask from nitrogen metabolism, okay. Then another simple topic is plant tissue culture, okay. From plant tissue culture you will get 2 to 3 questions. So on an average, minimum 10 to 12 questions, minimum 10 to 12 questions we will get from these slides, okay. From today's revision class, minimum 10 to 12 questions we will get out of 100, okay. So you have to be listen properly, okay. If you have any doubt, kindly ask in the chat section. I will reply, okay. Be attentive in class, okay. First, we are going to see techniques, major techniques in biochemistry and biotechnology, okay. We are, see, before going to reading any techniques, I want to give some of the hints for you, okay. First, for each and every technique, you have to know the principle, okay. On what basis the technique is working, okay. This is the most important thing. And what are its classifications, okay. These two are the most important thing you have to learn in each and every technique. And what is its major application, okay. We are dividing into three things. One is principle, second most important thing is application, and third most important thing is why we are doing this one, okay. This we are going to discuss for each and every technique, okay. We are going to start with electrophoresis, okay. Electrophoresis, the name itself is there. Electro means what? It's current. Phoresis means separation, okay. So electrophoresis is a technique that will used for separating the charged molecules under the influence of an electric field, okay. This is the general definition of electrophoresis, okay. So separation of charged particles under the influence of an electric field is known as electrophoresis, okay. If you see, DNA molecule is there, the DNA molecule will have phosphate groups. This phosphate will have negative charge, correct? So this negative charge can move towards the positive charge anode, okay. So this is the basic principle. If we apply an electric field, ion of opposite charge will move to its opposite electrode, okay. So this is the basic principle. So electrophoresis means it's a technique used for separation of charged particle under the influence of an electric field, okay. So the majorly the electrophoresis is used for what? For the separation of proteins, DNA and nucleic acid. This is another important point. We cannot use electrophoresis for separation of lipids, okay. This is another important thing. So we will use for separation of proteins, DNA. That means DNA and RNA, nucleic acid, okay. What is the major principle of this electrophoresis? We know the charged particles will migrate towards its opposite electrode. This we have already seen. This is the major principle. So the migration of the molecule will be determined based on the size, charge and shape of the molecule. If a molecule is small in size, that will move faster through the gel matrix, okay. We will use gel matrices, okay. In that gel matrix we will load our sample, okay. Okay, this we are going to see what is mean by gel, everything we are going to see, okay. This is the major components of an electrophoresis unit. Okay, major component of electrophoresis unit. If you see here, first a power supply will be there. Okay, power supply will be there. This will provide the electric current. Okay, this will provide electric current. The samples we will load in the gel. This is the gel molecule. This is the gel. The samples we will load in the well of the gel. Okay, this gel is made up of two different components. Okay, either it may be a agarose gel or it may be a polyacrylamide gel. This is most important thing. Okay, most of the DNA molecules are separated using agarose gel. For protein separation, majorly polyacrylamide gel will be used. Another objective point, important point also. Okay, apart from that, buffer system will be there, electrophoresis chamber will be there, and sample loading wells will be there. These are the major component of electrophoresis system. Okay. Next, there are two different types of electrophoresis are there. Okay. First is electrophoresis for protein and second is electrophoresis for DNA and RNA molecule, that is nucleic acid. These are the different types of techniques used for separation of proteins. Okay, these are the different types of techniques used for separation of nucleic acid. Here, the question what they will ask means, what is the different technique used for protein separation and what is the basic principle? On what basis the molecule are resolved or separated? This is the question they will ask. Okay, you see, there are different techniques are there. For example, if you see SDS page, SDS means what? Sodium dodecyl sulfide polyacrylamide gel electrophoresis. Okay. Here, what we will do means, our protein molecule will be there. This we will treat with SDS molecule. SDS is an anionic detergent. This is another important objective point. Okay. It's an anionic detergent. So, this will lead to formation of negative charge on the protein molecules. Okay. Negative charge on the protein molecule. So, the protein will be separated in the gel only based on molecular weight. This is the principle. Okay. In SDS page, the proteins will be separated only based on molecular weight. Here we are using anionic detergent, that is sodium dodecyl sulfate. Clear? So, this is the most important objective question they are asking repeatedly. Okay. But if you want to separate the proteins on its native stage without denaturating the protein, we can use native polyacrylamide gel electrophoresis. Okay, native polyacrylamide gel electrophoresis. The most important thing is here we will not use this anionic detergent, we will not use. In spite of this, we will use Zwitter ionic detergent. Okay, Zwitter ionic detergents we will use. This will not break the secondary and tertiary structure of the protein. So, the native structure of the protein will remain as it is. Okay. Another most important thing is isoelectric focusing. Isoelectric focusing. Here, the proteins will be separated based on the isoelectric point. Okay. The proteins will be separated based on isoelectric point. This you have to know. Isoelectric point means what? The pH at which the mobility of the protein is 0. Okay, mobility of the protein is 0. Okay, for example, this is the pH range. For example, 3 to 10 is there. If my protein have net charge of 0 at pH 6.1 means this is the PA, that is isoelectric point. Okay, this is the, for each and every protein there will be a characteristics PI. So, based on the PI, that means isoelectric point, we will resolve the proteins. Okay, this is clear. This is most important one. Another most important thing is two-dimensional gel electrophoresis, that is 2D page. Okay, the name itself is there. 2D page. Okay, 2D page. There are two dimensions are there. First dimension will be based on isoelectric focusing and second dimension will be based on molecular weight, that is SDS page. This is the repeated question they are asking in statement. Okay. Assertion and reason and statement question they are asking repeatedly. In two-dimensional polyacrylamide gel electrophoresis, the first dimension will be based on isoelectric focusing and second dimension will be based on what? SDS page, that is based on molecular weight, the proteins will be resolved. Okay, these are the different techniques used for separation of proteins. I hope this is clear for everyone. And for separation of nucleic acid, we will use agarose gel electrophoresis and capillary gel electrophoresis. Okay, so two different techniques we will use for separation of nucleic acid, agarose gel electrophoresis and capillary gel electrophoresis. I hope this is clear for everyone. Okay, this is the basic arrangement of the electrophoresis unit. If you see, first the sample we will load in sample well. Okay, then we will apply electric field. Because of that, ion of opposite charge will go to interact with opposite charge electrodes. Okay, because of this, the molecules will be resolved. This we will stained with ETBR or whatever chemicals we need for staining. Then we will determine the molecules. Okay, clear. So this is the basic steps in electrophoresis. Now we are going to see about the application. Okay, what is the application of this electrophoresis? First and most important thing is what? Protein purification we can do. For example, I have 10 proteins are there. If I know molecular weight of some of the protein, I can purify that one. So protein purification I can do. Molecular weight of a molecule I can determine by loading the markers also along with them. Then western blotting preparation, southern blotting and northern blotting preparations we can do. For example, in western blotting we will determine the proteins. Okay. First, the protein has to be resolved on gel. Okay, that we will done using electrophoresis. And in sudden blotting, DNA molecules will be separated. Okay, in northern blotting, RNA molecules will be determined. So, this is the basic. First, we will do the electrophoresis. Then, we will go for this northern blotting, western blotting and southern blotting. I hope the concept is clear for everyone. What is electrophoresis? What is the basic principle? And what is the application? And what are the different types? Clear? Just let me know, clear? Yes. If clear means next technique we can go. Okay. Next, yeah. Next we are going to discuss about centrifugation. Okay. Centrifugation, another important thing. We are going to discuss what is most important for exam point of view. Okay. First, centrifugation means what? It's a technique used to separate particles suspended in a liquid medium. Okay. It's a technique used for separation of the particle. If 10 different molecules are there, that will be dissolved in a medium, liquid medium. I will apply the centrifugal force. Because of the centrifugal force, the component will be separated into different segments. Okay. So whatever the molecule having higher molecular weight, that will settle first. Okay. Whatever having the less molecular weight, that will settle at last. Okay. So based on size, shape and density, we will apply the centrifugal force. Because of that, the molecule will be separated. Okay. This is another important point. Heavier or denser particle moves outward. That means it will settle first. Then lighter particles will settle at last. Okay. Then this is the most repeated objective question they are asking. 1 RCF is equal to 1 RCF is equal to how much RPM? Okay. 1 RCF is equal to 1.18 into 10 to the power minus 5 into R into RPM square. You just remember this one. This is a derivation. Okay. RCF means relative centrifugal force. This is a derivation. We have briefly discussed about this derivation in our lecture classes. Now we are just going to revise. So 1 RCF is equal to 1.118 into 10 to the power minus 5 into R into RPM square. You just remember this one. Okay. Next, another most important thing, if a molecule is sedimenting, it will take some time to sediment. For example, 1 second it will take or else 2 seconds it will take. Okay. So the sedimentation of the molecule can be determined or expressed based on Swedberg unit. Okay. Swedberg unit. Okay. 1 Swedberg unit is 10 power minus 13 second. This is another repeated objective question. Okay. 1 Swedberg means 10 power minus 13 second. That means if, for example, ribosomes are there, 30s subunit is there, 50s subunit is there. If you see this 50s means what? This is larger subunit, correct? This larger subunit will require less time for sedimentation. Okay, less time for sedimentation. So 50 into 10 to the power minus 13 second it will take to sediment. Clear? If you see 30s, 30s means it will require some more time for sedimentation. It requires 30 into 10 to the power minus 13 second. That means, that's why we are saying 30s, 50s. Clear? This is clear. This is another important point. Okay. So higher S value means it will faster sediment. Okay. This is another important point you have to remember. Okay. The centrifugation can be classified into two different types. Centrifugation can be classified into two different types. One is differential centrifugation, another is density gradient centrifugation. Differential centrifugation and density gradient centrifugation. Okay. In differential centrifugation, what we will do means, you just remember the medium we are using will have an uniform concentration. Okay. Uniform concentration will be there. So the molecules will be separated based on size and mass. But in density gradient centrifugation, what you will use means, the medium will have different density. Okay, we will use sucrose or glycerol or other cesium chloride like that chemicals to generate the centrifugation with different density of the medium. Okay, from higher density to lower density like that it will be there. Okay, so the density of the medium will be varied. This is the basic difference between differential centrifugation and density gradient centrifugation. Okay, further the density gradient centrifugation is classified into two types. This is another repeated objective question. Okay, further, the density gradient centrifugation is classified into two types: rate zonal centrifugation and isopycnic centrifugation. You have to just remember only one point. Okay, only one point in this. See, okay. This isopycnic centrifugation is independent of time. It is independent of time. This is the most important objective questions they are asking repeatedly. But rate zonal centrifugation will be depend on time. Okay? The centrifugation and the sedimentation of the particle will be independent of time in isopycnic centrifugation. Okay? This is another important point you have to remember. I hope the centrifugation concept is clear for everyone. Now we are going to see about spectroscopy. Okay? Spectroscopy is different, spectrophotometer is different. First we are going to see what is spectroscopy, then we are going to see about spectrophotometer. See, listen properly, each and every point is very, very important in techniques in biochemistry and biotech. You have any doubt means kindly let me know there itself. Okay. Now we are going to see what is meant by spectroscopy. Okay. Spectroscopy means what? If a molecule is there, it can interact with the light. If a light will interact with the matter or any molecule, the light can be what it can be do. Light can be reflected or absorbed or transmitted like that. Different phenomena will happen. Okay. So spectroscopy means what? It's an interaction between electromagnetic radiation and then matter or molecule. Okay. Why that interaction is happening? Based on the interaction, what we will do means, we can identify the component. What is this component X? And we can also measure the concentration of the component and the molecular structure of the analyte also we can determine. Okay, so the basic principle of spectroscopy is what? If an electromagnetic radiation will interact with matter, some of the light will be absorbed, transmitted or reflected. This is the basic principle. So this is spectroscopy. Okay. The instrument used for this spectroscopy is known as spectrophotometer. Clear? A spectrophotometer is an instrument used to measure the amount of light absorbed by a particular wavelength. Okay, absorbed by the sample at particular wavelength. Okay, the basic principle of spectrophotometer is what? Beer-Lambert's law. This is another important objective question. Okay, Beer-Lambert's law. Okay, we can divide into two parts. So Beer law means what? Beer law means absorption of the light will be directly proportional to concentration of the analyte. Clear? Absorption is directly proportional to concentration. What is meant by Lambert's law? Lambert's law will say that will directly proportional to path length. Clear? Path length. So, Beer-Lambert's law is the basic principle for spectrophotometer. Okay. Clear? So, we have seen now what is spectroscopy and what is the principle of spectroscopy. Clear? I hope this is clear for everyone. Now we are going to see what are the different classification of spectrophotometer. Okay, this is the basic arrangement you see. First light source will be there. Okay, monochromator will be there. Why we are keeping monochromator means each molecule or different type of molecule will observe light at different wavelength. To select a particular wavelength from this light, we will use monochromator. Okay, in cuvette we will keep our sample. So the incident light will fall on these molecules. I0 is incident light. So some of the light will be absorbed by the analyte in the molecule. So whatever the light that is remaining, that will be passed here. So the final is I. So I0 by I will give the value of the light that is particularly absorbed by our analyte in the sample. Okay, analyte in the sample. So we will get an absorbance value. For example, A equal to 0.260. This is the absorption of the particular analyte in the sample. So based on the absorbance, we can calculate the concentration by using standard graphs. I hope this is clear. This is very simple concept only. You have to apply it. Okay. Now we are going to see the classification of spectrophotometer. Okay. I told already the radiation or the electromagnetic radiation will interact with the matter that will cause light to absorb, scatter or reflect. Okay. So these are the different types. Okay. If you see the types, there are three different types are there. Okay. One is absorption spectroscopy. That means what? The light will be absorbed. Okay. So substance will absorb specific wavelength of light. So how much of light is absorbed? That is directly proportional to concentration of the analyte. Okay, clear. So example of absorption spectrophotometry is what? UV visible spectrophotometry, IR spectrophotometry, and AAS spectroscopy. This is very important. Okay, the examples are important. They will ask what is the example of absorption spectroscopy. Okay, statement and reason question they will ask. Okay, next thing, emission spectroscopy. Okay, if a molecule is there, if light is passing on that, this molecule will go to excited state. From excited state to coming to ground state, it will emit a photon of light. Okay, this is known as emission phenomena, correct? Emission phenomena. So what is the amount of light emitted will be directly proportional to the concentration of analyte in the molecule. So the basic principle is what? Atom or molecule absorb energy first and become excited. When they return to ground state, they emit light of specific wavelength. This emission will be directly proportional to concentration of the analyte. So these are the example of emission spectroscopy: flame, fluorescence and atomic spectroscopy. Clear? Flame, fluorescence and atomic spectroscopy. Another most important thing is scattering. Light can be scattered from the molecule, so we can say that as scattering spectroscopy. Okay, scattering spectroscopy. The typical example for scattering spectroscopy is Raman spectroscopy and dynamic light scattering. This is most repeated question and most important question also. Okay, Raman spectroscopy and dynamic light scattering is an example of scattering spectroscopy. Okay, then another most important objective I want to say here. Okay, if you want to determine secondary structure of proteins, we can use circular dichorism. Circular dichorism spectroscopy we can use. Okay, if you want to determine the tertiary structure of a molecule, we can use XRD, X-ray crystallography we can use. Okay, NMR we can use, nuclear magnetic resonance, NMR we can use. Okay, cryo-EM, cryo-electron microscopy we can use. These are the different techniques we are using for determining the three-dimensional structure of the molecule. This is repeated question they are asking. Okay. So secondary structure means circular dichorism, tertiary structure means XRD, NMR and cryo-EM we can use. Okay. This is the basic difference. This we have already discussed properly. Absorption spectroscopy, emission spectroscopy and scattering spectroscopy we have discussed. Okay. These are the different wavelength. On what wavelength the molecules are observed. Okay. These are the different wavelength. Gamma rays, X-rays, UV, visible, infrared, micro and radio waves are there. Okay. This is the wavelength range just I have given for your thing. So UV visible spectroscopy means the light source we are giving is in the range of UV and visible range. Then most important thing you have to remember is radio waves will be used. Radio waves will be used in nuclear magnetic resonance spectroscopy. This is another important thing. Okay. Radio waves are used in nuclear magnetic resonance spectroscopy. Another important thing, if you want to find the functional group in a molecule, we will use infrared spectroscopy, infrared or IR spectroscopy. Okay, clear. I hope this is clear for everyone. Clear. Whether you are listening, clear. Kindly let me know whether it's clear. If it's boring, kindly let me know. I know this techniques in biochemistry is one of the vast topic. Yes. Clear. Yes. Very good. Very good. Very good. Okay. Now we have seen the classification of spectroscopy. Absorption, scattering, okay, emission, like that we have seen. Now we are going to see two major example for spectroscopy. This is the most repeated question they are asking every year. Okay. So atomic absorption spectroscopy and ICP-OES. Okay. ICP-OES. We are going to see what is what. Okay. What are the important point in this atomic absorption spectroscopy, ICP-OES and MALDI-TOF. Okay, clear. So atomic absorption spectroscopy, the name itself it's there. It's a type of absorption spectroscopy. Okay, it's a type of absorption spectroscopy. Then which is absorbing the energy? Atoms are absorbing the light energy. Okay, that's why we are saying atomic absorption spectroscopy. Simple. Okay. Here what we will do means we will take our sample, then our sample will be converted into atoms. This each atoms will absorb light at specific energy. So the amount of light absorbed is directly proportional to the concentration of the particular analyte. Okay. So how we are converting the molecule to atoms? This is the key question they are asking. So how we are converting the molecules to atoms? Okay. So the molecules to atoms will be converted by two different ways. Okay. One by flame ionization, another by graphite furnace. Okay. So the sample will be converted into free atoms by two different ways. One is flame ionization and the second is using graphite furnace. This is the question they are asking repeatedly. Okay. The similar thing, AAS we are using for multiple element can be analyzed at a time. Okay. The similar thing we are using for ICP-OS. ICP-OS expansion, this they have asked in 2019 JRF. Okay. JRF 2019 they asked. Okay. ICP-OS means what? Inductively coupled plasma optical emission spectroscopy. It's a type of emission spectroscopy. Okay. You have to see why we are saying inductively coupled plasma. Okay. So this each and every word having meaning. Okay. Simple thing. In atomic absorption spectroscopy, molecules are converted into atoms by what? Flame ionization they have did or graphite furnace they have used. Here what they have used means they will use argon atoms along with the radio frequency waves to generate high plasma. Plasma means what? It's an high heat temperature molecules will be generated. Because of this high heat, the molecules will come here. This will be converted into different atoms. Clear? So, different atoms will be formed. Okay. It's a type of emission spectroscopy. So, light will be observed. It will go to excited state. From excited state, it will travel to ground state. While coming to the ground state, it will emit a photon of light that will be proportional to the concentration of the analyte. I hope this is clear for everyone. These two are very important topic. Okay, clear? Any doubts in electrophoresis, centrifugation and spectroscopy? Any doubts? Any doubts? Kindly let me know. If you want break also, kindly let me know. Yeah, no doubts. Yes. Safura, no doubts. Surbi, no doubts. Yes. Okay. Okay. Three most important topics we have covered. Today next topic is blotting. Okay. Blotting. So blotting, this is another important technique. Okay. In blotting, you have to know three important things. Okay. What are the different types of blotting and for what purpose we are using? Okay. There are three major types of blotting techniques are there. First, we have to know what is meant by blotting. What is mean by blotting? First you have to know. So blotting means what? Is a technique used to transfer biomolecules from gel matrix into a membrane for detection and analysis. That means what? In first slide itself I told electrophoresis we will do, molecules will be separated. This we will transfer to membranes. Okay. So just we are blotting on the membranes. Okay. Nitrocellulose membrane. Okay, we will blot these molecules on nitrocellulose membrane. This is known as blotting. Blottical means it can be either DNA molecule, RNA molecule or protein molecule that we will transfer to the nitrocellulose membrane for detection. What we are going to detect? In southern blotting means we will detect the specific DNA molecule. This is the important thing. Southern, okay, DNA molecule. Okay, southern is for DNA molecule. Okay, southern for DNA molecule. Okay, northern blotting for RNA molecule. Western blotting we can also say as protein blotting or immunoblotting. Okay, protein or immunoblotting. This is the technique used for detection of protein in our sample. This is the key important point you have to remember in blotting. Okay, here we have listed out what is southern, northern and western blotting. What are the different procedures they are using. This is not important. The most important thing you have to remember is how the double stranded DNA of a molecule of DNA will be converted into single stranded before blotting to the membrane. Okay. So you see here the double stranded DNA molecule will be converted into single stranded molecule by alkaline hydrolysis. First is alkaline hydrolysis they will do, and most important thing, after converting or after fixing the molecule into the nitrocellulose membrane, they will do baking, okay, baking at 80 degrees Celsius for fixing the DNA molecule to the membrane. This is the two important extra points you have to remember. Okay. First, alkaline treatment they will do. Okay. So the double stranded molecules will be converted into single stranded molecule, or else they will use deep urination method. Because of that, the double strand will be converted into single strand. Another most important thing, the single stranded molecule will be fixed on the membrane by baking or UV cross-linking. These are the different techniques. Okay. Now we have completed the blotting. The major difference we have given here, you just go through once. Okay. Already we have covered briefly in our classes. Okay. The another most important thing is MALDI-TOF. MALDI-TOF. Here what question they are asking means, what is the expansion? Like ICP we have found inductive coupled plasma optical emission spectroscopy. Like the expansion for MALDI-TOF, matrix assisted laser desorption ionization time of flight. Okay, the each and every word having meanings. Okay, matrix assisted laser desorption ionization time of flight. Here the question what they will ask means, what is the principle of MALDI-TOF? Okay, the basic principle of MALDI-TOF is the molecules will be separated based on mass to charge ratio. So not only based on mass or not only based on charge, mass to charge ratio. This is the most important objective question. Mass to charge ratio. Okay. Now we will just briefly revise the principle. Okay. You see sample preparation we can see. This is the matrix. Why we are saying matrix assisted? So here some matrix will be there. Okay, this matrix, for example, synapic acid matrix molecule will be there. Here we'll have a chromophore that can absorb the light energy. Okay. Here I am adding my sample molecule. Okay. If I place a laser on this, the molecule will be converted into ions. Clear? The molecule is converted into ions. See here, different ions are there. Okay. Ions of different molecular weight will be there. Okay. These ions will be accelerated with applying an high voltage. Okay, so this will be accelerated by applying high voltage. Then this will pass in an high vacuum environment. Okay, for example, I have one iron ball is there, one wooden ball is there. Okay, if I need more amount of energy to throw that iron ball, but I need only less amount of energy to throw that wooden ball. So the wooden ball can reach the detector very easily, correct? So the time of flight will be very much less for lesser weight ions. This is the question they are asking. Okay. So if a molecule having a lesser weight, that will reach the detector soon. If a molecule having high weight, that will reach the detector at last. Okay, so this is the principle. Based on mass to charge ratio, the molecules will be determined in MALDI TOF. That's why we are saying time of flight, the time taken to reach the detector by an analyte or an molecule. Okay, clear? I hope this is clear for everyone. Okay, now we are going to see the most important techniques. We are going to just brush up. These are very basic concept. I know you will write the answers if they ask this question. We are just going to revise. Okay, this revision is most important for your preparation. Okay, we are going to revise now. Okay, so PCR means what? Polymerase chain reaction. This is the polymerase chain reaction. For the discovery of PCR, Carey Mullis got Nobel Prize. Okay, Carey Mullis got Nobel Prize for the discovery of PCR. Okay. So PCR, why we have to use PCR? We know in our cell DNA replication will happen. Okay. DNA replication means what? Faithful copies of the DNA will be produced. Okay. But in vitro condition also we can synthesize the DNA by using template and specific primers. By using thermostable DNA polymerase enzymes. Okay. That is tag polymerase that was isolated from Thermus aquatrophicus, tag polymerase. This will have high thermal stability. Even under 95 degrees Celsius also, the protein will not degrade and the enzymatic activity will be conserved. This enzyme we can use for doing the PCR reaction. Okay, this is clear. I hope this is clear. Okay, so the basic component of PCR, what it will be there? Template DNA will be there. Primers we will add. Then tag polymerase enzyme we will add. DNP buffer and magnesium chloride we will add. Okay. The basic principle is what? See, you just see here. Okay. Our template DNA will be there. If we keep at 90 to 98 degrees Celsius, it will convert it into single strand molecule. Then our primer will go and bind here. After that, extension will be happened. Extension temperature is most important. 72 degrees Celsius is the optimum temperature for extension. Okay. So a single molecule has been converted into two molecules. Okay. Like that, exponential multiplication will be happen. The question what they will ask from this PCR means, okay, how to find the final concentration of the molecule? If I am taking only one molecule, if I am taking only one DNA molecule at the end of 10 cycle, at the end of 10 cycle, what is the number of molecule present there? Simple formula is there. N0 into N, 2 power N. Okay, this is the formula. Okay. So 2 power N is number of cycle. N0 is the initial concentration of the molecule. For example, 100 micromolar concentration is there. 100 nanogram concentration is there into 2 power 10. 10 is 10 cycle is there. So exponential multiplication will be happened in PCR. I hope this is clear. And this is 2019 JRF question. How to determine the primer temperature of melting point? Okay. How to calculate the TM of the primers? Okay. This is the formula. If you want to calculate the TM of an primer, formula is 2 into A plus T plus 4 into G plus C. I hope this is clear. 2 into A plus T plus 4 into G plus C. This we have already discussed. Okay. Now variants, you have to just see the variants. We are not going to deeply see the concept, just what are the different variants. Okay. RT-PCR. RT-PCR means what? Reverse transcriptase polymerase PCR reactions. qPCR or real-time PCR means what? Here the expression of the genes can be determined. Apart from that, nested PCR, hot star, touchdown, inverse, like that, different variants are there. You have to remember this qPCR. qPCR we can here this qPCR can be used for determining the differential expression of the genes. Okay, differential expression of the genes can be determined. Okay, differential expression of the genes can be determined. This also we have already seen. To know the final concentration of the molecule, we have to multiply the N0, that is initial concentration of the DNA template, multiply by 2 power N. N is number of cycle. Like these problems they can ask. Okay, like these problems they can ask. Clear? Clear? Any doubts is there? Any doubts is there in PCR? This is last technique, chromatography. Okay. Last technique we are going to see chromatography. Any doubts so far? I know this is very vast topic. Any doubts? Yes. Safura, no doubts. Any other persons? Yes. Surabhi, no doubt. Yes. Yes. Now we are going to see about chromatography. Okay. This is the last technique class. Okay. Chromatography. After that we will start plant tissue culture. Don't worry. Okay. This will take just 10 minutes. Huh? Chromatography. Why we have to do chromatography? Okay. See, the basic principle of chromatography is it's a separation technique. If I have a different molecules, I want to separate a particular analyte from that, I can use chromatography. So, chromatography is a technique, that is a separation techniques in which components are separated based on differential distribution between stationary phase and mobile phase. First, we have to know what is meant by stationary phase and what is meant by mobile phase. See, in chromatography, two phases will be there. Stationary phase, another is mobile phase. Okay. Stationary phase, that means, for example, it is a column. Okay, it will not mobile in nature. Then mobile phase will be there. This what it will do means it will carry our sample or analyte to the stationary phase. Clear? So our mobile phase, what it will do means our sample will be there, that will be taken and given to the stationary phase. Okay. Here itself, in the definition itself, it's there. Based on the differential distribution between the stationary and mobile phase, our molecule will be separated. For example, if the molecule is highly soluble in mobile phase, it can soluble here, then it will go here, it can elute first. Clear? So, how the matter, how the analyte is interacting between stationary phase and mobile phase, based on this, our molecule can be separated. Okay. There are different techniques are there. You have to know these are the different techniques. From here, sure, I will assure you now itself, from chromatography, every year they are asking two questions. At least one question every year. Okay. So chromatography, there are different principles are there: adsorption chromatography, partition and charge based, size based and affinity based. We are going to see just the principle. Okay. We are going to see just the principle of each and every technique. Okay. You see here, different techniques are there: adsorption, partition, ion exchange, size exclusion and affinity. This is the repeated question. Ion exchange chromatography is the repeated question they are asking. Okay. So adsorption chromatography means based on differential adsorption, we will separate our molecule. Okay. Size exclusion, then ion exchange means based on charge or based on molecular size. Okay. You just see. Okay. So adsorption chromatography, this we have already discussed. If a molecule having strongly absorbed to molecule, it will move slowly. Okay. So simple thing. If in our column, our molecule will attach more tightly. This is our column. In our column, if the molecule attached more tightly, it will take more time to elute from this. Okay. If it is less tightly absorbed, it can elute first. This is the basic principle. Okay. Next partition, based on different partition we will be doing. Okay. Next is what? Next is ion exchange chromatography. Okay. Ion exchange chromatography. Here two different types are there: cation exchange chromatography and anion exchange chromatography. Okay. In cation exchange chromatography, okay, different resins we will use based on the interaction of the molecules. Okay. The analyte will be separated. Okay. Another most important thing, okay, size exclusion chromatography. Here, based on the size, the molecule will be separated. Just we are going to revise only. Okay. Then another last technique is affinity chromatography. In affinity chromatography, we know antigen have different affinity with antibody. Enzymes will bind with its substrate. Receptors will bind with ligands. Based on the interaction between the ligand and the molecule, different things will be separated. Okay. Another thing is HPLC and UPLC. This is very important concept. You know these things. This I don't want to revise also. In HPLC, we will use high pressures. Okay, high pressure we will use. Okay, high pressure we can use. Okay, in GC, gas chromatography, the compound should be volatile in nature. Okay, so volatile compounds will be there. Okay, so the compound will be converted into volatile in nature, then it will be resolved. This is the difference between liquid chromatography and gas chromatography. Okay, with this we are completing our techniques in biochemistry. Okay, we have completed the major techniques in biochemistry. Okay, the last important topic is what are the different techniques used for DNA protein interaction, and what are different technique used for DNA RNA interaction and RNA RNA interaction. This we have already discussed in last class itself, yesterday class itself. You just see once. Okay, this we have already discussed in our last class. Okay, the most important is RNA protein interaction. This we have already seen. RNA immunoprecipitation, clip RNA pull down, EMSA, Y3H. These are the different techniques used for studying the RNA protein interaction. Okay, with this we are completing the techniques in biochemistry. We can have a break of 5 minutes. Okay, I will see you at 11:55. Okay, I will see you all at 11:55. Okay, Lokendra, sir, you know Sairam, sir. Kindly ask the doubts in biotechnology only. TK, personal messages you can message in WhatsApp. Kindly ask the doubts in subject only. TK, I will see you at 11:55. 11:55. Yes, thank you. Whether I'm audible now? Yeah. Yes. Fine. Fine. Now it's fine. Just let me know now it's fine. Kindly, kindly reply students whether I'm audible now. Yes, we have completed techniques in biochemistry and biotechnology. Okay, now we are going to see briefly about plant tissue culture. Okay, plant tissue culture. See, from plant tissue culture, sure you will get 3 to 4 questions. So every year, if you analyze the previous year question paper, what are the questions they are asking? First, you have to know what are the questions, in which type of questions they are asking. From history, from history of plant tissue culture, sure they are asking one question. So you have to focus much on history of plant tissue culture. From here there, 1 or 2 questions you will get. And second most important thing, what are the different cultures, what are the different techniques, cell culture techniques they are using? For example, anther culture, pollen culture, like that different techniques are there. From that culture they are asking. Then most important thing, protoplast fusion, protoplast fusions, somatic embryogenesis. From this portion they are asking every year one question. Okay, so these are the most important topics we have to cover in plant tissue culture. Okay. The term biotechnology, was given by Carl Lerke. This is just for your information. Okay. Who is known as father of plant tissue culture? Okay. Father of plant tissue culture is Gottlieb Haberland, or G. Haberland is the father of plant tissue culture. This is very important objective point. Okay. G. Haberland is the father of plant tissue culture. Okay. Now we are going to see the history only. We are going to see history only. Okay. First, Hanning. Okay. Hanning has given the Hanning has discovered embryo culture. This is JRF 2019 question. Okay. JRF 2019 question. Hanning, embryo culture was given by Hanning. Okay. Another important thing, Robinson and Cotti has given the root tip culture. This is another important point. Okay. Cotti and Robinson has given the root tip culture. Just you go through once. Okay. From here you will get question. Okay. Whatever the important point that I am going to discuss. Okay. Skoog. Skoog has discovered that adenine stimulate the cell division. Skoog discovered adenine is important for cell division. Okay. Skoog and Thucyd discovered adenine induces bud formation. Then most important thing, the term embryogenesis was given by F.T. Stewart. This is most important objective question. Stewart has given the name embryogenesis. Okay, another most important question. Okay. Then 2022 question. This is JRF 2022 question. Meristem tip culture. What is the purpose of meristem tip culture? If you want to produce virus-free plants, we can use meristem tip culture for virus elimination. This is another important point. Who has given the meristem tip culture means Morrell and Martin. Morrell and Martin. So, meristem tip culture for virus elimination, this was discovered by Morrell and Martin. These are the objective questions. Okay. Then protoplast isolation. There are different techniques for protoplast isolation. Enzymatic method and mechanical method. Okay. Who has given enzymatic method? Who has given the mechanical method? Okay. The mechanical method for protoplast isolation was given by Von Klerker. This is another important point. Cocking has given the enzymatic method of protoplast isolation. Okay, Cocking has given the enzymatic method of protoplast isolation. Whatever the point I am saying, you just have to revise that one only. Don't go deep into the concept. Just you revise. Klecker for mechanical method, Cockling for enzymatic method. Okay, don't waste the time. Okay. Then Guha and Maheshwari. Guha and Maheshwari for haploid production, haploid plant production. Guha and Maheshwari for haploid production. Okay. Then another most important thing, this we have discussed in yesterday class itself. Paul Berg first created the recombinant DNA molecule from SV40 and lambda virus. Okay. Stanley Cohen and Herbert Boyer, they have developed the first GMO organization. This also we have found GMO organism first they have done. Stanley and Cohen Herbert. This we have discovered yesterday itself. Okay. Now we are going to see the most important topics, most important definitions also. They can ask what is mean by totipotency, what is mean by D-differentiation, what is mean by D-differentiation, what is explained and what is callus. Okay, you have to know. Okay, so what is mean by plant tissue culture? Plant tissue culture means what? It's an in vitro cultivation of plants, seeds, plant plots or plant pots on nutrient medium under aseptic condition. Controlled sterilized condition. Okay, we will in vitro condition, we will cultivate the plants or parts of the plant to develop the entire plant under aseptic condition. Okay, so whatever the plant pot we are using, that we will say as explants. Okay, that are explants. Then callus means what? This is most important thing. Callus is an unorganized or growing mass or group of undifferentiated cells. Okay, this is most important definition that forms an explant. Okay, so callus is very important and this is the most repeated objective question they are asking. Callus is an heterogeneous. It will be there in heterogeneous. Callus will be a heterogeneous molecule. This question they have asked in our objective like in PYQ also they have asked this question. Okay. Now we are going to see the three most important point. Okay. Sir, we are taking only the leaf or seed. How from there the entire plant are developing in vitro condition? Why it is happening means because of the totipotency. Okay. You have to know what is mean by totipotency. Totipotency means what? The phenomenon of an de-differentiation and redifferentiation are inherent in the capacity of the plant that give rise to whole plant. There are two different concepts are there, de-differentiation and redifferentiation. Because of that, the single explant will be converted into an entire plant. Okay. Now we are going to see what is de-differentiation and what is re-differentiation. Okay. So de-differentiation means mature cell. For example, leaf I am taking, reverting to meristematic state and forming undifferentiated callus. That means leaf is converting into callus. This is undifferentiated mass of cell. Then this callus is converting to whole plant. This process is known as redifferentiation. That means the ability of competent cell of callus to differentiate into an whole plant. Clear? So the de-differentiation and de-differentiation is the basic for totipotency. I hope this is clear for everyone. These are the definition they are going to ask in the question paper. Okay. So we have seen. Okay. The another most important thing I already told, the explants will be grown in a growth medium. Okay, growth medium. What are the different types of culture? What are the composition of the medium? That also you have to know. Okay. So the nutritional need of the culture will be similar to inter plants. Okay, there are different types of cultures will be there. Callus culture, cell culture, organ or protoplast culture will be there. These are the different types of culture. And the medium is majorly composed of what? Mineral salts will be there, carbon sources will be there. Apart from that, vitamins, growth regulators, and organic supplements will be there. This is just for your understanding. Okay. What are the different sources of vitamins they are using? Growth regulator. For example, auxin we can use, cytokinin we can use. The ratio will determine the root and shoot formation. This also we have seen. The root for cytokinin and auxin ratio, if equal concentration is there, means callus formation will be there. If cytokinin concentration is more, that will lead to formation of shoot. If auxin concentration is more, that will lead to formation of root. So based on the concentration of these plant hormones, we can develop rooting and shooting from our explant. This we have already discussed. Okay. Now we are going to see the different media types. Okay. What are the common medias they are using in tissue culture? Okay. The most and commonly used media is MS media. Okay. Then followed by Gamborg or B5 medium. Okay. This is originally designed for callus and suspension culture. For callus and suspension culture, Gamborg or B5 media will be used. Okay, clear? And the most important thing you have to remember, suspension culture will be primarily used for production of secondary metabolites. Okay, secondary metabolites can be produced by using suspension culture. I am going to tell what are the important points only. Just you focus the important points. These are the important and key points. Okay. Now we are going to see the sterilization techniques. I already told aseptic condition should be there first thing. So how we can sterilize? For example, different glassware we have to sterilize, media we have to sterilize, hormones we have to sterilize, explains everything we have to sterilize. That then only the aseptic condition can be maintained. Okay. This sterilization techniques they have asked in match the following. Okay. They have asked in match the following. They have given the different sterilization technique: steam sterilization, dry sterilization, filter sterilization, and UV sterilization. These are the different techniques. Okay. Steam sterilization, dry sterilization, and filter sterilization and UV sterilization. Here, what question they asked means steam sterilization will be used for what purpose? Okay. So media will be done by steam sterilization. We will use autoclave. What is the temperature and pressure we are using in autoclave? This is another important objective. Okay. We will use 121 degrees Celsius for 15 psi for 20 minutes. This is the temperature, pressure and time used for steam sterilization in autoclave. And the second most important thing is dry sterilization. Here we will use water oven. What we will sterilize here? We will sterilize glasswares and metallic instruments. Okay. This is the important point. Filter sterilization. We are using different plant hormones. The plant hormones will be heat liable. So that should be filtered using filter sterilization. Okay. Then UV sterilization. UV sterilization by what? UV irradiation we can use. Okay. UV irradiation we can use. Then most important thing, We will use laminar airflow chamber. Okay, laminar airflow chamber that will create an aseptic condition for doing our plant tissue culture works. Okay, this laminar airflow chamber we will use HEPA filter, high efficiency particulate analyte filter. The efficiency of this HEPA filter is 99.97%. This is another important objective question. Okay, HEPA filter we will use the then the HEPA filter efficiency will be there 99.97%. I hope this is clear for everyone. These are the different sterilization techniques. Now, how we can sterilize our explant? How we can sterilize our explant? The explant can be sterilized by using sodium hypochlorite or calcium hypochlorite solutions we can use for sterilizing the explant. I hope this is clear how we are sterilizing the different things. Okay. Now we are going to see the different types of culture. Okay. You have to know what are the different types of cultures are there. Okay. Embryo culture is there, seed culture, meristem culture, cell culture, callus, bud culture, organ culture, and protoplast culture. These are the different culture. So the name was given based on the explant what we are using for culturing. Okay, whether bud or seed, meristem or protoplast. Based on that, they have given the name. I hope this is clear. Okay. Then most important thing, artificial or synthetic seed. Here what questions they will ask. Okay. First we will see what is meant by artificial seeds and what are the questions they will ask. Okay. Artificial seed means what? It's a synthetic structures made by coating the somatic embryos. Okay. With a protective matrix to mimic the natural seeds. Questions what they are asking repeatedly means what is the protective matrix or encapsulating matrix used in synthesizing of artificial or synthetic seed. Okay. They will use sodium alginate. This is the hydrogel they are commonly used. Okay. Sodium alginate. This is the most important objective question you have to remember. Okay. Sodium alginate. This will be primarily used for synthetic seed production. Okay. Now we are coming to another most important thing, haploids. Okay. We are going to see what is haploid, what are the different techniques used for haploid production, who is the scientist discovered this haploid culture. Okay. We already seen Guha and Maheshwari has discovered this haploid culture. First we will see what is meant by haploid because they have asked the definition of haploids one time. Okay. So haploids are plants that contain single set of chromosome that is known as gametophytic chromosome number will be there. Gametophytic chromosome number will be there. Okay. So this is the definition for haploid. Okay. So naturally how the haploids will be produced in natural I am saying. In natural that may be produced due to apomixis or parthenogenesis or apogamy. These are the different phenomena that will lead to haploid production in natural condition. But if you want to develop an haploids under in vitro culture, there are different techniques are there. Anther culture is there and oval culture is there. Okay. Now we are going to see how we are doing the anther culture, how we are doing the oval culture, okay, and what are the media we are using in that. Okay. We are going to see the important points only. Okay. You see, we already discussed Guha and Maheshwari. Guha and Maheshwari was the person who was first discovered this haploid culture. Okay, haploid culture. Okay. There are two different cultures of their androgenesis and gynogenesis. Okay. In androgenesis, we will use microspores or male gametes. In gynogenesis, ovary or ovule will be cultured. Ovary or ovule will be cultured. Okay. This is just for your understanding only. Okay. Now we have now how to double the chromosome. Okay. How to double the chromosome or chromosome doubling how we will do. Haploid we already discussed is a set of only one set of chromosome will be there. That is gametophytic chromosome will be there. How we will convert this into 2n? So chromosome doubling. How we will do? This is the most important thing. Okay. And mechanism also you have to know. So chromosome doubling. So chromosome doubling will be primarily done by what? Chromosome doubling will be primarily done by colchicine treatment. This is very important objective question. Colchicine treatment we will do. This colchicine treatment, what it will do means, it will inhibit the spindle apparatus or spindle fiber formation. Because of that, chromosome doubling will be happen. Okay. Then to determine the ploidy level, how to determine the ploidy level? Just I have given here. You just go through once. Okay, this is not that much of important. Just you go through once. How we can determine the ploidy level. Okay, this also we have already discussed. Colchicine treatment we can use, or else endomitosis we can use for chromosome doubling. Okay, or diploidation of the chromosome can be done. Okay, next most important thing, protoplast and protoplast culture. Protoplast and protoplast culture. Okay, first we will know what is meant by protoplast. Okay, protoplast means what? A cell without the cell wall is known as protoplast. Protoplast means what? A cell without a cell wall is called as protoplast. Okay. The term protoplast was given by Einstein. This is JRF 2020 question. Okay. The term protoplast was given by Einstein. Okay. And the isolation for Klecker, this we already discussed. Mechanical method of protoplast isolation, Von Klecker. Enzymatic method by Cockling. They have attempted to isolate the protoplast from the intact leaf molecules, from intact leaves. Okay. How to isolate the protoplast by using mechanical and enzymatic methods. Okay. I already told there are two different methods are there. Mechanical method and enzymatic method. The question what they will ask here means, what are the different enzymes and what are the source of enzyme they will use for enzymatic method of protoplast isolation. Okay. This is the question they are going to ask. Okay. This is the question they are going to ask. You just see here. Okay. What are the different enzymes they will use? We will use cellulase, hemicellulase, pectinase. Okay. This these are the component of the cell wall. For degrading this, we will use cellulase, hemicellulase, and pectinase we will use. Okay. These are the different sources of the enzyme. This slide is very, very, very important. Okay. You have to know what are the different sources of enzyme. Cellulase, hemicellulase, pectinase is also known as macerozymes. Okay. Then drysellase, glucellase, and others. This example and the source from where they have identified. This is the objective question they have asked one time. Okay. So these are the different enzymes. For example, hemicellulase source is what? Rhizozyme HP. Okay. Pectinase or macerozyme from Rhizopus fungus. Okay. Trichoderma viridi or Trichoderma resi is the primary source for cellulase. Okay, this is the question they are going to ask. So you have to be very clear about this one. Okay. Then protoplast viability test. If I have isolated the protoplast, how I can check whether the protoplast is viable or not? So simple thing, we will stain the protoplast with different chemicals. Okay. We will do staining the protoplast with different chemicals. Okay. In this, what question they will ask means, what are the stains that will stain only the dead protoplast? Okay. There are two different stains are there. One is spinosapharine. This will stain only dead protoplast. And another most important thing is Evans blue exclusion dye. The live cells will be excluded, only the dead cells will absorb it. I hope this is clear. So these are the different staining techniques. Okay. The last concept is somatic hybridization. If I have isolated one protoplast from one species and I have isolated protoplast from another species, I can fuse this protoplast 1 and 2 to form a somatic hybrids. That is the purpose of somatic hybridization. Okay, somatic hybridization. What is the purpose? Sexual incompatibility or incompatibility barriers can be overcome. This is the primary aim of this somatic embryogenesis. Okay, somatic embryogenesis. What happened with the during fusion? Nucleoplasm and cytoplasm will be come together and it will form a somatic hybrids. Okay. Then what is mean by hybrids? This they can ask. What is mean by hybrids? If only nuclear genome of one parent is retained, but cytoplasmic gene from both parents are fused, means that is known as hybrids. I hope this is clear. So Nuclear genome of one parent or one plant will be there, but the cytoplasm of both the gene, but the both the species will be there in that. That are known as hybrids. Okay, this is another important objective question. Any doubt is there in plant tissue culture? We have completed plant tissue culture. I hope the concepts are clear for everyone. Any doubts? This is highly sufficient to cover plant tissue culture. Any doubts is there? Kindly let me know. Any doubts? Surabhi, no doubt. Safura, any doubt? Adarsh, Lokendra, yes. If no doubts means we are going to start nitrogen metabolism. Okay. We are going to start nitrogen metabolism. This is another important topic. I already told minimum 3 to 4 questions they will ask. Minimum 2 questions they will ask from nitrogen metabolism. Okay. We are going to see now what is the importance of nitrogen in the plant. Okay. Then we are going to see about nitrogen cycle, how the nitrogen cycle is happening. Then what are the different processes? Denitrification, nitrification, biological nitrogen fixation. What is the importance? Okay, how the enzymes are regulated. From A to Z, we are going to discuss in this nitrogen metabolism. Okay, you don't want to go and read in some other books. Okay, whatever I have given in the slides, these are highly sufficient. I have compiled the notes from standard books. Okay, you don't want to go and read these topics in any other books. Okay, so listen the class properly. Okay, we know the importance of nitrogen in the plant. Okay, what it will the most important thing? It's an constituent of amino acid and proteins, nucleic acid, chlorophyll, and it is essential for plant growth and development. These are the most important function of nitrogen. Okay, apart from that, it is important for coenzyme and cofactor formation. It will increase the yield and quality. Also involved in the synthesis of secondary metabolites like alkaloid synthesis. Okay, okay, these are the basic importance of nitrogen in plant. Okay, before going to see the basics about nitrogen metabolism, first I want to say some of the definitions. Okay, some of the definition in nitrogen metabolism. Okay, this definitions they are asking in statement question. Okay, statement question they will ask whether statement 1 is correct or statement 2 is wrong, like that statement question they will ask. Okay, listen properly. Okay, if I am applying nitrate fertilizer in soil, for example, this is soil. I am applying nitrate fertilizer. Okay, nitrate fertilizer I will apply. This nitrate fertilizer will be uptaken by the plant. Okay, plant roots will be there, it will uptake. Okay, this nitrate will be converted into ammonia or amino acid. From this ammonia will be converted into amino acid. Okay, so the process of converting nitrate to ammonia. Okay, nitrate to ammonia. What is the process? We are going to see. What is the process of converting nitrate to ammonia? Then what is the process of converting this ammonia to amino acids? Okay, this also we have to see now. Okay, we are going to see now. The applied nitrate can be converted into nitrogen gas and it can escape from the soil. What is this process? This we are going to see. Okay, how the nitrogen in the atmosphere will be fixed in the soil as ammonia. Okay, biological nitrogen fixation. This process also we are going to see. So there are different or multiple processes are there. Okay, first we are going to see the basic definition of each and every process. Basic definition we are going to see, and we are going to see the deep about each and every concept. Okay, first definition. Okay, first definition is what? What is meant by nitrification? Okay, nitrification means what? In the process of converting ammonia into nitrite and nitrate. Okay, so ammonia will be converted into nitrite. This nitrate will be converted into nitrate. This is the process of nitrification. So denitrification means what? The process of converting nitrate into nitrogen gas. Okay, this is known as the nitrification. Then ammonification means what? Conversion of organic nitrogen into ammonia. Okay. Then mineralization means what? Release of inorganic nutrient from organic matters. Okay. These definitions are very important. Okay. Then biological nitrogen fixation, it's the conversion of atmospheric nitrogen into ammonia by microbes. This is the most important definitions you have to remember. Okay. Now we are going to see about nitrate assimilation. This is the repeated question, nitrate assimilation. You just listen properly. Okay. We are going to see about nitrate assimilation. Okay. Nitrate assimilation. If we apply nitrogenous fertilizer like urea we are applying, okay, how the nitrate is uptaken by the plant, how in the plant the nitrate is converted into ammonia, and how it is incorporating into the amino acid. This we are going to see step by step. Okay. For example, we are applying the nitrate fertilizer in the soil. This will be enter into the root cells by nitrate transporters. Okay. Nitrate transporters will be there. Okay. So this nitrate transporter help in taking the nitrate from the soil. Okay. Once it reaching into the root cells, okay, there are two different enzymes are there. One is nitrate reductase, NITRAT, nitrate reductase. What this will do means it will convert the nitrate into nitrite. Okay. It will convert nitrate into nitrite. This will be present in cytoplasm. You see here in cytoplasm, nitrate reductase is there, converting the NO3 minus to NO2 minus. Here it is utilizing NADH as an electron donor. Okay. Electron source is from NADH. Okay. So it converted into nitrate. So this nitrate will be converted into ammonia by NIR, that is nitrite reductase enzyme. This will be present in plastids. This is the question they are asking repeatedly. Okay. In root cell, NIR will be present in leucoplast, but in shoot or mesophyll cells, it will be present in chloroplast. Another most important differentiation you have to know, the electron source for NIR enzyme in leucoplast is NADPH, but the electron source here is reduced ferredoxin. This is the electron source. Okay. Then most important thing, nitrate assimilation will happen in both root and shoot cell. This is the another important objective question they have asked in JRF 2023. Okay, nitrate assimilation will happen in both root and shoot cells. This is clear. I hope this is clear for everyone. Okay. Now we are going to see the structure of the enzymes. Okay. The structure of the enzyme. What is the structure of this nitrate reductase? What is the structure of this nitrite reductase? Okay. If you see the NR enzyme, we are going to say hereafter NR. NR will have three different domains will be there. FAD domain, cytochrome B55 summon domain, and molybdenum containing cofactor domain. Okay, molybdenum containing cofactor domain. So three domains are there for converting NO3 minus to NO2 minus. The electron source will coming from NADH. This is another important point. Okay. So this enzyme will have three different domains. It will be present in cytoplasm. These are the three different domains. Electron source will be NADPH, NADH. Okay. This is the important point you have to remember. Okay. Another important thing, NIR. If you see the structure of nitrate reductase, that is NIR. Okay. It will have iron sulfur clusters. If you see this diagram, this also have three different domains. First is iron sulfur domain will be there. Apart from that, FAD domain will be there. And cirroheme will be there. This is the question they are asked. JRF 2024 they asked this question. Cirroheme containing enzyme is what? That is NIR, nitrite reductase. And the source of electron Source of electron for NIR is reduced ferredoxin in chloroplast, NADPH in leucoplast of root. Leucoplast of roots. Okay, clear? Plastid and leucoplast of root. This is the important questions. Okay, they will ask, you have to remember. So, what is the localization of the enzyme? What is the structure of enzyme? What is the electron source of the enzyme? What is the substrate and what is its product is forming? Okay, now we are going to see if nitrate is converted into nitrate, nitrate is converted into ammonia. How this ammonia is incorporated or converted into amino acid? This will be done by ammonia assimilation. Ammonia assimilation will be done by GS-GO-GAT pathway. GS-GO-GAT pathway. So here we are going to see what is GS. GS means glutamine synthetase. Okay, glutamine synthetase. GO-GAT means what? Glutamine 2-oxoglutarate aminotransferase. This we have already discussed in our class. Or else we can also say as glutamate synthase. Glutamate synthase. You just revise once. This is the key question they are going to ask. Okay. So the ammonia will be formed by the action of GS enzyme. It will convert it to glutamine. This glutamine will be converted into two molecules of glutamate by the action of GO-GAT. Okay, by the action of GO-GAT, amino acids will be formed. This amino acid will be act as an amino donor for synthesis of other amino acid. I hope this is clear. So GS GO-GAT pathway will be happen. Okay. Now we are going to see the inhibitors of this GS GO-GAT pathway. You just see here only. Okay. So glutamate structural analog is glufosinate. Okay, glufosinate. This is an herbicide. Okay, if you apply this glufosinate herbicide, it will go and bind here. So the GS enzyme will be inhibited. Okay, if you use azacirine, azacirine, if you use azacirine, this will go and bind with GO-GAT enzyme. Okay, clear. So GS enzyme will be inhibited by glufosinate. Azacirine will be inhibited. Azacirine will act as an inhibitor for GO-GAT enzyme. These are the key objective points you have to remember. Okay, you see the structure here. Glufosinate, azacirine. Okay, glufosinate will inhibit the enzyme GS. Azacirine will inhibit the enzyme GO-GAT. Okay. Now we are going to see the regulation. This is also another important point. Okay. Now we are going to see regulation of this nitrate reductase enzyme. Okay. Here what question they asked means what are the different regulation mechanisms involved in NR enzyme. They have given this in assertion, reason and questions. Okay. You see here first the gene expression of this NR will be regulated by different components. Nitrate, light and carbohydrate source will act as a positive regulator of converting the NR gene to NR enzyme or NR protein synthesis will be happened. Okay. This NR enzyme, if it is phosphorylated at serine residue, it will become inactive. Clear? So, if the NR enzyme is in phosphorylated form, NR phosphorylated form means it will be an inactive molecule. It is an inactive molecule. If NR is present in dephosphorylated form, that will be an active enzyme. Okay. Then another important thing what they have asked means once the NR is phosphorylated, here 14-3-3 protein will come and bind. Okay, 14-3-3 protein will come and bind. That will further inhibit the enzyme activity of NR. This question they asked in JRF 2024. Okay, so NR enzyme will be inhibited by phosphorylation and further the binding of 14-3-3 protein will inhibit the NR activity. This is the important regulation of nitrate reductase. Okay. Then we know this amino acids will have carbon sources along with that amino sources. The amino sources will be given by nitrate assimilation and from where the carbon sources will come. The carbon sources will be primarily coming from CO2 assimilation process. Okay, carbon source will primarily come from CO2 assimilation process. Okay. Now we are going to see how the different amino acids are synthesized. Okay, you just have to remember once there are 6 major classes of amino acids. Okay, based on the biosynthetic precursor. This we are going to see in our metabolism class also. Okay, you just see once alpha ketoglutarate family, 3-phosphoglycerate family, oxaloacetate family, pyruvate family, PEP family, and ribose phosphate family. These are the 6 families. Okay, we are going to see very briefly about each and every class in our upcoming metabolism revision class. Okay, any doubt is there in nitrate assimilation? Any doubt is there? Any doubt? No doubt. Very good. Shall we proceed or you need break, students? If you need break means we can take 5 minutes break. Break you need? Kindly let me know. Surabshi, Safura, others, you need break or shall we proceed? No, no break. Okay. Okay, students, now we have completed the nitrate assimilation process. Nitrate assimilation process we have completed. Now we are going to see about denitrification and nitrification process. Okay, we are going to see in denitrification what are the important points you have to remember. Okay, so first we are going to see the definition of denitrification. We know denitrification means what is a microbial process of converting the nitrate into nitrite. Okay, nitrate into nitrite. This is the overall reaction. Nitrate will be converted into nitrite. Nitrate will be converted into nitric oxide. Nitric oxide will be further converted into nitrous oxide. Then the nitrous oxide will be converted into nitrogen gas. If you see here, it's a reductive process, correct? The oxygen has to be removed here. It's a reductive process. So the final product forming is what? Nitrogen gas. Nitrogen gas. So in some cases, nitric oxide will be formed up to here. So nitric oxide will form. This will act as a greenhouse gas. Okay, this will act as a greenhouse gas. So now we have seen what is meant by denitrification, what is the product formed, okay, what is the significance. Okay, these are the different, these are the conditions required for denitrification process. First thing, anaerobic condition or low oxygen condition should be there. Yes. Waterlogging soil should be there. This also we have seen. And most important thing, the bacterium. So it is a microbial process. The bacterium involved will be majorly facultative anaerobic bacteria. Okay, facultative anaerobic bacteria. Okay, this is another important point. It is a facultative anaerobic bacteria. These are the typical examples for denitrifying microorganisms. Okay, Pseudomonas denitrificans, Bacillus denitrificans, Paracoccus, Thiobacillus, and Micrococcus denitrification. These are the typical example for denitrification microorganism. This they have asked one time in question. Okay. Now we are going to see the importance and harmful effect of denitrification. Importance of denitrification, this we already know. Nitrogen cycle can be maintained. Okay. If excess nitrate is there in soil, that is nitrate toxicity will be there. That can be removed by denitrification process. Soil nitrogen balance can be maintained. Environmental importance also is there, then wastewater treatment also is an beneficial. What is the negativity or what is the harmful effect of denitrification? Most important thing, fertilizer loss will be there. If more denitrification is there means the applied nitrogen fertilizer loss will be there. Then another important thing, greenhouse gas production will be there. This also we have seen. Greenhouse gas production will be there. Nitric oxide will be formed. This we have already seen. Okay. Now we are going to see about nitrification. Okay. We have completed about denitrification. Now we are going to see about nitrification. Okay. So nitrification means what? It's the process of converting ammonia into nitrate. Yes, ammonia into nitrate. This nitrate will be converted into nitrate. This is the overall reaction for nitrification. This is very, very important point. Okay. Now we are going to see what are the conditions required and what are the steps. Okay. This is the steps, you know. First step is what? Ammonia will be converted into nitrate by the action of enzyme ammonia oxidizing bacteria. These examples are very, very important. Okay. Examples are very, very important. Ammonia oxidizing bacteria is what? Nitrosomonas, Nitrococcus and Nitrospira. Nitrosomonas, Nitrococcus and Nitrospira. And nitrate oxidizing bacteria is what? Nitrobacter and Nitrococcus. Okay. This is confusing, but they are going to ask question from this one only. This one only. What are the example of ammonia oxidizing bacteria and nitrite oxidizing bacteria? This is the question they are going to ask. TK students, you have to remember that properly. Then what are the important? Same thing only. Nitrogen cycle will be there. Most important, soil fertility can be enhanced. Wastewater treatment will be there. These are the key importance. Okay. Harmful effects of nitrification is what? Nitrate leaching will be there. This we have already seen. Soil acidification will be there. Apart from that, indirect nitrogen loss will be there. Okay. Okay. These are the disadvantage of nitrification. Okay. The last topic of today's section, we are going to see about biological nitrogen fixation. Okay. We can have a break for 3 minutes. Okay. I will see you all at 12:42. Okay. We can have a break. Then we will continue this biological nitrogen fixation. Okay, students. Yes, shall we proceed? Biological nitrogen fixation. Yes, shall we proceed? Yes, last topic. Okay. Biological nitrogen fixation. Now we are going to see the definition. Okay. Biological nitrogen fixation means what? How the molecular nitrogen gas in atmosphere is converting into ammonia with the help of bacterium species. Okay. So biological nitrogen fixation is the process by which atmospheric nitrogen is converted into ammonia by certain microorganism using nitrogenase enzyme. So the enzyme involved in biological nitrogen fixation is nitrogenase enzyme. Okay. The see, even though the atmospheric nitrogen have around 78% of atmospheric nitrogen is there, if you see the N triple bond N will be there. If you see the nitrogen gas, triple bond will be there. This is highly impossible to break by normal things. That's why the biological nitrogen fixation is there. Okay, this triple bond will be converted into ammonia. Okay, this will be broken and converted to ammonia. Because of this, more amount of energy will be utilized. Okay, more amount of energy will be utilized. This is another important and objective question, repeated question. Okay, overall reaction of biological nitrogen fixation. If you see, for converting each molecule of nitrogen to 2 molecule of ammonia, we need 16 molecules of ATP. This is another important point. Okay, 16 ATP is required for converting a molecule of nitrogen gas to 2 molecule of ammonia. Another important thing, 8 electrons are required. This is also another important thing. 8 electrons are required for converting the nitrogen into ammonia gas. Okay. The nitrogen complex, first we have to see about the enzyme complex. Okay. I already told nitrogenase is the enzyme that will be involved in conversion of this nitrogen gas into ammonia. Okay. This nitrogenase enzyme have two different components. We can say two different complex will be there. Component 2 and component 1. Okay. Component 1 we can also called as iron molybdenum iron protein. This is component 1. Okay. Component 1 is iron molybdenum protein component and component 2 is iron protein complex. This is very important. Okay. So component 1 is molybdenum iron protein complex and component 2 is iron protein complex. Okay. The component 1 is made up of alpha 2 beta 2 structure, that is heterotetramer. Okay. Heterotetramer will be there. Okay. Component 2 is made up of homodimer. Homodimer. This is the question they have asked one time. Okay. Alpha 2 structure will be there. Okay. Alpha 2, alpha 2 and beta 2. This is the question they have asked. Then another most important thing what they asked means, what are the genes that will code this alpha and beta subunit of component 1 and what are the genes that will code the alpha subunit of component 2. Okay. NIF-D and NIF-K. DK, TK. D and K will code the alpha and beta subunit of molybdenum iron protein. Okay. NIF-DK. So NIF-H, NIF-H for component 2, that is iron protein component. This is clear. So what are the genes that will code molybdenum iron protein and what are the genes that will code the iron protein component? This is very important and you have to remember component 1 is a molybdenum iron protein, component 2 is iron protein. They can ask what is the cofactor for the nitrogenase enzyme. Okay, it's a molybdenum containing enzyme, so molybdenum will be act as a cofactor. Okay, then if you see the major characteristics of nitrogenase, this nitrogenase enzyme are highly sensitive to oxygen. This is JRF 2022 question. Assertion and reason they have asked. Okay. It is a highly sensitive to oxygen enzyme. It requires large amount of ATP. That is 16 ATPs are required. And most important thing, it will be present only in nitrogen fixing organism. Nitrogen fixing organism. Okay. This is the just diagram for your understanding. Okay. And the most important thing, this nitrogenase we will call as sluggish enzyme. Why we are saying this as a sluggish enzyme? I already told there are two different components are there. Component 1 and component 2. Okay. For every electron transfer, the each component has to be separated and has to be reassembled. Okay. So it will time consuming also. It is sluggish enzyme. That's why we are saying it's a sluggish enzyme. For every electron transfer, the two component has to be separate and it has to be rejoined. That's why we are calling this as an sluggish enzyme. Okay. So if we see total 8 electron has to be passed, so 8 times the component will be separated and it will join. And another most important thing, for every electron transfer, 2 molecules of ATP will be hydrolyzed. Okay, 2 molecule of ATP will be hydrolyzed. Okay. There are 3 different types of biological nitrogen fixation are there. This is another important thing. 3 types are there. One is symbiotic, another is non-symbiotic, third is associative symbiotic. Here what questions they will ask means, what are the example of symbiotic nitrogen fixation bacteria? Okay. What are the example of non-symbiotic or free living bacteria? And what is the example of associative symbiotic bacteria? Okay. We know symbiotic nitrogen fixation bacteria, rhizobium is the typical example. Okay. Non-symbiotic or free living, this is most important thing. Okay. Acetobacter is an example, Clostridium and Cyanobacter. Okay. Associative symbiotic means Azospirillum with grass and cereals. Okay. This is the example from here. Sure, you will get one question. What are the examples? Example of symbiotic, non-symbiotic and associative symbiotic bacteria. Okay. From here, we are going to get one question. Okay. Now we are going to see the mechanism, how the symbiotic nitrogen fixation will be happened. Okay. You just imagine why the root nodules has to be formed in pulses. Why it has to form? Okay. Then another most important thing. This is a bacterium. Why that plant? Why the pulse species is not recognizing that bacteria as an foreign molecules? Okay. It's an see. This is an interesting thing. If you see, why the nitrogen fixation has to be happen. So we know these pulses, this pulse plant will provide the carbon source for the bacterium. This bacteria will fix the nitrogen to ammonia. It will give to the plants. Okay. So if you see, first thing, the rhizobium will be present in the rhizosphere region of the soil of the root. Okay. So first the plant will release the flavonoids compound. Okay. That will act as a signaling molecule. This flavonoids will act as a signaling molecule, so the rhizobium will come and near to the plant. Okay. Once it come and near to the plant, root hair curling will happen. Okay. So root hair curling will happen. Then infection thread formation will happen. Okay. Through this infection thread, the bacteria will enter into the plant cell. Okay. Once it enter into the plant cell, it will form an symbiosome membrane or peribacterial space. Okay, it will form a symbiosis membrane. Inside that, the bacteria will colonize. Okay, here it will convert the nitrogen to ammonia. These are the basic step, nitrogen to ammonia. Now we have to remember in which source the bacteria is giving the nitrogen to plants, and in which source the bacteria is obtaining the carbon source from the host organism. This is another important point. Okay, you have to remember one thing. Okay, you have to remember one thing. The nitrogen will be given to the plants in the form of ammonical source, particularly amines. Glutamine, amino acids will be given. Okay, the bacteria will take the carbon source in the form of malate. Okay, that is dicarboxylic acid, malate. Okay, you just remember. Okay, that is there in next slide. Okay, the bacteria will take malate as the carbon source. Nitrogen will be fixed here. In glutamines will be, that means amide form of nitrogen will be given to the plants. Okay, and another most important thing, I already told the characteristics feature of this nitrogenase enzyme. It is highly sensitive to oxygenase enzyme. Okay, but the rhizobium requires oxygen. It is an aerobic bacterium. Okay, which component is protecting the nitrogenase sensitivity towards the oxygen? This they may ask. Okay, so there comes the protein leg hemoglobin. Okay, leg hemoglobin is the important protein that will prevent nitrogenase enzyme from oxygenase damage. Okay, another most important thing, it will maintain low oxygen for respiration in the rhizobium. Okay, I hope this is clear for everyone. Okay, I hope this is clear for everyone. Okay, what are the major factors involved in biological nitrogen fixation? This is the last thing. Okay, oxygen, if high oxygen is there means nitrogenous activity will be inhibited. Okay, then different things, soil pH, carbon assimilation rate, everything they have given here. Just you go through once. This is the most important thing. Oxygen is more means the activity of nitrogenous will be reduced. Okay. Then another most important thing, then last topic of today's lecture. Okay. We know this is the overall reaction for converting the nitrogen to ammonia in bacterium. Okay. If you see here, 8 molecules of H plus will be taken here, but only 6 molecules of H plus will be incorporated in ammonia, but 2 molecules of H2 will be released as it is as hydrogen. Okay. This hydrogen releasing is decreasing the efficiency of this nitrogenous enzyme. In some of the strains, there is an enzyme known as uptake hydrogenase. Uptake hydrogenase. What it will do means, it will convert this hydrogen. It will convert this hydrogen into 2 molecules of H plus and 2 molecules of electron. How the conversion of this H plus and electron will improve the efficiency of this nitrogen fixation? That we are going to see. This is the last topic. Okay. If you observe here properly, 2 molecules of H plus are there and 2 molecules of electron will be released. If you see, these electrons will be used for energy conservation. That electron can flow in electron transport chain. Because of that, ATP production can be happen. This produced ATP can be utilized for nitrogen fixation. I hope this is clear. This is the first point. Then second most important thing, it will improve the plant growth also. It will reduce the hydrogen loss. Okay, these are the important thing. Then most important thing, energy conservation will be there. Then another important thing, the ferredoxin, reduced ferredoxin formation. For this also, electrons will be utilized. Whatever the electron will be synthesized from this step, whatever electron is synthesized, that can be used to convert the oxidized ferredoxin to reduced ferredoxin. Okay, these are the importance of uptake hydrogenase in plant. Okay, these are the importance of uptake hydrogenase. Okay, with this we are completing our lecture for today's lecture. Okay, any doubts are there? We have briefly discussed about plant tissue culture, techniques in biochemistry and biotechnology, and nitrogen metabolism. Okay, any doubts is there? Kindly let me know any doubts are there. No doubts. Okay. Any suggestions? Kindly drop about the quality of the class in comment section. That will be useful for improving the things. Okay. After this lecture, kindly post your valuable comments. Okay. If you have any doubts, you can type in the chat session. I will reply. Okay. Tomorrow we are going to see about photosynthesis, A to Z of photosynthesis. Phytohormones we will discuss. Okay. Tomorrow same timing, 11 to 1:30, we will discuss photosynthesis, phytohormones. Apart from that, some of the topics from plant physiology. Okay. Please, one marathon class for SRF. Yes, we are planning that one also. We will conduct Safura. Okay. Thank you, students. Thank you.